Okay, so the idea of this talk will be to explain you how you can use FCS to study, for example, the dynamics of certain molecules in the cell. So just to explain a little more what we do in the lab, let's imagine that you want to understand how a city is organized. So you can do two different types of experiments. The first one, for example, will be isolating a woman, like this one here, put her in an artificial context and just observe what she does. The other way of understanding this city will be, for example, observing the same or other women working, behaving, interacting in the real context. So the main idea of the experiments I will show you today will be observing processes in living cells to really understand the rules that govern this city that is the cell. So today, can you see the whole image? Okay. So today, the idea of the talk will be, instead of having a woman, here we will have the glucocorticoid receptor. I think Diego Pressman talked a lot about the glucocorticoid receptor a couple of days ago, and we will be using fluorescence fluctuation method to study certain aspects of this receptor. So Diego talked a lot about the glucocorticoid receptor. Do you remember something? I can see you, so you can answer me. Just say yes or no. No? Okay.
Well,
more or less. So, as you probably know, glucocorticoids are widely used to treat diseases involving the immune system. So there is a big interest in the area to understand how the glucocorticoid receptor works, which are the molecular mechanism involving all these responses. So what Diego probably showed, I don't know if you can see, there is a cell in here. The receptor normally distributes in the cytoplasm, and whenever you stimulate the cell, for example, with an artificial glucocorticoid or a natural one, the receptor translocates into the nucleus, and in the nucleus, it works as a transcription factor. One important thing that I'm gonna perhaps talk today is that in the nucleus, the receptor can form these multiple foci, and of course, if I have time, I will talk just a little about that. Okay, so today I will try to show you how FCS can be used in certain biological problems. We are gonna address two different questions. The first one is if we can observe the receptor in action, and the second is if we can dissect interactions between the receptor and other biomolecules that are relevant to its function. So just to simplify the talk, I will talk about the wild-type version of the receptor. This is the DNA binding domain, the ligand binding domain. You must remember, or perhaps you don't remember, that Diego explained that the receptor, when it is active, it forms dimers. And we will compare the behavior of this receptor with a monomeric version of the receptor that has 2-point mutations, one in the GBT, the other in the LBT. And, well, the functional output of GR-MON, as it is named, this mutant, is completely different from the wild-type receptor. Of course, we studied many mutants, but for pedagogical reasons, I will explain only our results in these two variants of the receptor. Okay, so the first question is, okay, let me see if I can hide this. Okay. So the first question is, can we observe the receptor in action? And one of the ways to answer this question is by using FCS very rapidly. Here you have, for example, a confocal microscope. You focus the laser on a cell, let's say in this spot, and as Enrico explained, by doing so, you determine or you define a very small, a tiny observation volume. And every time a fluorescent molecule comes in and out of this observation volume, it will, well, it's in the dark state. Whenever it comes in, it will start to fluoresce until moving out. So if you collect intensity as a function of time with a time scale in microseconds, what you are going to detect are fluctuations due to these molecules that move in and out of the confocal. And Okay, what Enrico also says is that, for example, the duration of this fluctuation will be related to how fast these molecules move in and out of the confocal bond. Relevantly, let's imagine, for example, if you have binding sites in the volume you are focusing the laser, molecules move in, they will stay a little, some period of time attached to the targets and then move out. So now this intensity fluctuation will contain information on this binding. For example, if the molecules stay for a long period of time bound to the targets, the fluctuation will last longer, and consequently, they will contain information on the binding process. How do you extract the information regarding the fluctuation? Well, Enrico showed you also that you can do that through the autocorrelation function. This is intensity fluctuation at time t. This is intensity fluctuation at time t plus a lag period of time tau, and you do this calculation. Essentially, or qualitatively, what you do is the following. You have in here the fluctuation intensity trace for short tau periods. What you do is multiply the red one with the blue one. The blue one is exactly the same as the red one. So if tau is really, really small, you do this multiplication and you get a high value of the correlation function. So what you are measuring is the similarity between the intensity trace and itself but shifted a given period of time tau. So if tau increase, this self-similarity decrease, and whenever tau is high, this multiplication, the average of all the multiplication will give 0, okay? So if now you have faster molecules, this fluctuation will last for a shorter period of time, and the correlation, the self-similarity will decay faster than before, okay? So the autocorrelation function contains information on the dynamics of the molecules. In very simple system, this is, for example, one of the work, a previous work we did in my lab. Here you have, for example, fluorescent molecule attached to a lipid bilayer. So the fluorescent molecules diffuse in 2D. You can fit the experimental data with an equation that accounts for the motion mechanism. In this case, two-dimensional diffusion. By fitting this data, you can, for example, obtain the diffusion coefficient of the molecules you are interested in. Okay? Going back to the top, what happened with a glucocorticoid receptor or other transcription factors? We ran some simple FCS experiments in the nucleus of cells. You can see here, here you can see the control cells that were not stimulated, and in blue, you can see cells that are stimulated. And you can see that the correlation curve shifts to higher values of tau when you stimulate the cells. So the question is, can we obtain quantitative information on what is going on with the receptor in these conditions? Of course, we cannot fit this data with a random diffusion model because we know that the receptor is binding to chromatin. Yeah. So I'm gonna do like a parenthesis here. Some years ago, we were studying other transcription factors, in fact, the pluripotency transcription factors in living embryos. We obtained a correlation course very similar to the one I showed you. And by doing control experiments that I don't have time to show you today, and some simulation, we could determine that the correlation course of this transcription factor can be interpreted considering a model in which the TF bind to DNA in two different time scales, long-lived and short-lived interactions. With this model, in fact, this model is contained in this equation. You can derive this equation from this model. This equation fits correctly the experimental data. And the nice thing about this equation is that you can recover two relevant parameters of the body. The first one is the residence time, which represents the time the molecule, the transcription factor spent bound to the chromatin, either short-lived or long-lived site. And the second parameter is the fraction bound, the bound fraction, sorry, which is the relative amount of transcription factor that are attaching to either short or long-lived sites. So by doing FCS, we can recover quantitative parameters on the binding in living cells. So for example, in the case of the GR, here are the same curves I showed you before. And when you analyze the data, you can see that after stimulating the cells with X, both the residence time and the bound fraction increase, indicating that the stimulation of the receptor promotes the binding of the receptor to a target site and for longer period of time. What happened with GR mutant, the monomeric mutant? You can see here that the parameters are more or less the same as those measured in the control inactive receptor. So what we can observe in here is that the interactions with chromatin of GR-MON are impaired. So these two mutations affect interactions of the receptor with DNA. Second question, can we dissect the interactions of the receptor with other biomolecules relevant to its function? And to answer this question, we use fluorescence correlation spectroscopy, cross-correlation spectroscopy. I think Enrico already explained this technique to you. Did him? Lionel?
Yes, yes.
Okay.
Yes, yes.
So very rapidly, here, for example, you have the receptor bound to a green dye. We're gonna analyze the interaction with the co-regulator, in fact, with the co-activator, which it's labeled in red, it's red fluorescence. So in the microscope, what we do is use two laser lines to excite both of them, and then we collect in independent detectors the green fluorescence and the red fluorescence. So in the green channel, what you detect is either the heterodimers, because they have the green label, or green monomers. And in the red channels, you will collect the heterodimers and also the red monomers. The nice thing of this type of experiment is that every time heterodimers move in and out the confocal volume, it will produce simultaneous fluctuations in both intensity traces. See, here you have simultaneous intensity in the green and red detectors. Here, the green monomer only produces fluctuation in the green and the red in the red. How do you extract information regarding this simultaneous fluctuation? Well, by analyzing the cross-correlation function that is defined very similar, as you can see, at the autocorrelation function, but you correlate the intensity trace in the green channel with that in the red channel. And this will be higher than 0 whenever you have these simultaneous fluctuations in both channels. So we did that with the GR and mCoA2, which is the coactivator, a relevant coactivator of the receptor. Here you can see the autocorrelation in the green channel, in the red channel, and the cross-correlation. You can see here that this cross-correlation has an amplitude higher than 0, showing that heterodimers between GRs and the coactivators are formed in this condition. By analyzing the amplitudes of these correlation curves, we can calculate an apparent association constant. Remember that we are doing all these calculations inside the living cells, inside the living nucleus. And what we observe is that whenever we stimulate the receptor, the apparent association constant increase, showing that more interactions between the receptor and the coactivators are produced. And the monomeric version of the receptor presents a lower association constant, showing that the mutations of this receptor impair the interactions with the coactivators. Okay? So second question that I will show you very fast is why the active GR distributes heterogeneously in the nucleus. Is this a general behavior of nuclear receptor? I will not show our last results on this topic, but this is something that passes the field of steroid receptor for a long period of time. Why is this important? Well, because if you imagine you have a given reaction, a biochemical reaction inside, for example, the nucleus, you have in here A plus B that gives C. If the reaction occurs in an homogeneous media, the speed of the reaction will be something. However, if molecules concentrate in a given region of the nucleus space, here, for example, you have more A and B, so you have a higher probability of encounters between molecules, and the velocity of the reaction will go up. However, if the reaction, for example, occurs outside this foci, because you need an enzyme that do not go to foci, as you concentrate the molecules here, the speed of the reaction will go down. So then I think about this heterogeneous distribution of molecules is that it is a different way of modulating the speed of the biochemical reactions inside the cell. And this is something very, very important in the field because most steroid receptors from this foci in the nucleus. We've been studying this during a long period of time. The overall idea is that by using FCS and line FCS or scanning FCS methodologies at the level of this foci, we could determine that for forming the foci, you need an initial interaction of the active receptor with certain regions of the chromatin. After these initial interactions, more molecules are recruited to this initial seed. Many of these molecules are multivalent, so they interact with many, many other molecules, finally forming the condensate. We could determine most of these steps by analyzing the dynamics at the level of the condensate. So the final idea of my talk is that whenever we think in transcriptional output, we focus on the specific interactions between the transcription factor, for example, and the target on the DNA. We now know that both dynamical interactions in the whole nuclear space and the heterogeneous spatial distribution of molecules affect this interaction, and consequently, it modifies the transcriptional output. And I think I am done with